Welcome to the Soilcraft Regen Agronomy Podcast. At Soilcraft, our mission is to innovate and lead in the field of regenerative agronomy, forging a path that empowers farmers to produce food that nourishes and heals both the planet and its inhabitants.
And I thought, oh man, that would be cool. It's a huge agricultural pest. Everybody knows about the two-spotted spider mite, and yet I don't really have a very strong background in mites. I mean, I guess I'm stronger than most due to the degrees, where we understand about mites, spiders, and scorpions, and even lobsters and crabs, because they're all arthropods to a certain extent. But now I got a chance to delve into the mites, and 1 year later, I mean, there were jaw-dropping moments during this process of looking at the two-spotted spider mite. So I sat down and I wrote a report on this, and I'm in the process of looking to see where this is going to go. But now I feel that I've got so much information about the two-spotted spider mite, and I have been able to determine with relative accuracy what it is keying in on, why it is doing what it's doing, based upon just looking at the frequencies that I was able to decipher.
So in your mind, that's incredible. In your mind, what is the purpose of mites and the two-spotted spider mite specifically after that much research? I mean, I can't think of anyone who would be more qualified to say, I think this is the purpose. I think this is why they're here and what they serve to do.
They have several purposes. One, they cannot clean out water that well, so they have to wait until things are dehydrated. And so was able to back this up. And so once dry conditions become relevant, the spider mite does well. It's always there. It's always doing what it's doing. You can find them in almost every agricultural field on the planet, but their numbers do not grow exponentially, literally exponentially, until dry conditions are met. So once the cell starts to dry up and the water is no longer a factor, it becomes considerably easier for them to suck out the contents of a plant. So that's the one thing. Go ahead, Trent.
I have a question really quick because I'm dealing with two-spotted spider mites in hops currently as we're speaking. And it's been my observation that utilizing cytoplasmic plant fluid analysis, i.e. SAP analysis, that there is a correlation to salt levels. And when I say salt, I'm specifically talking about chloride. Also, there seemed to be some correlation to ammonium in the plant sap and mite pressure, but I think it's more due to chloride. And so my question with what you just said is that could make sense. If the plants are in a stressed, hot, dry environment, they're respiring or perspiring rapidly because of the environmental temperature above ground, and they're uptaking moisture, and there's not enough carbon sink or other elements to balance out the available salt that plant is uptaking. The moisture leaves the leaves in respiration, and the salt accumulates, which then would make sense with what you just said, why we have a higher incidence of mite pressure and them replicating and becoming a major issue. Does any of that make sense?
It does. Yeah, it makes a lot of sense at all. I wasn't able to find a connection specifically with the chloride ion. However, having said that, we also understand when the chloride ion concentrations increase, the Brix levels go down. And Brix levels being a measure of plant health. So when we have high chloride levels, the plant is now compromised and the Brix levels go down. And there's a research paper out there which was finding that the two-spotted spider mite optimally was looking for plants between 1 to 2 Brix when it was, I think it was getting exact, like 1.6 to 2.2. They had some exact figure. This is not research that I have done, but this kind of gave us an idea of how, at what point, and not just that the plant is unhealthy, but it needs to be brought down to a certain point before the mite actually feeds, because we all know, and I think you guys know as well too, that the mites are ubiquitous. They're found in Washington as well as they're found in Florida and everywhere else. Yeah, I mean, it's a ubiquitous pest right now, and because of that, their numbers don't increase exponentially unless you have very particular situations that result. And so these are some of the situations that we're discussing with them. When these situations come about, their numbers increase dramatically, and it can be stopped almost immediately, just based upon knowing what they like and what they don't like. So the chloride items I haven't been able to make a connection to. However, as the chloride ions increase, the Brix levels are going to go down, which makes it more attractive to the mite, because obviously they're only going after an unhealthy plant. I mean, I've given the presentation about Brix and insects, and a lot of people are familiar with it, but I wasn't talking about mites during that presentation because I didn't have that information at the time. But now that I've got enough of a background, I could, safely say that yes, the two-spotted spider mite is also going after plants that are unhealthy, just as the insects do. And so if I had to remake that particular YouTube presentation, I would remake it and include some of that updated information, which I'm now updating with Soilcraft and the two of you right now, and anybody who happens to be listening as well.
So with what you said about the mites needing a more dehydrated environment and not being able to deal with the water or evacuate the water, could you speak a little more to that? And with that, is there the possibility that if a plant is elevated in a specific salt, potentially such as chloride, that it would indicate that it's somewhat dehydrated?
It would, because usually salt leads to that dehydrated state, and usually because we're putting down high salt fertilizers and sometimes even high salt pesticides. And so, yes, that can easily contribute to it, because salt, the most common salt is NaCl, sodium chloride. So when you have excess sodium, because the sodium and the chloride disassociate, you're also going to have an excess of chloride. So that goes with what you were just saying. Yes, there is an association there.
Yeah, but oddly enough, we will see the accumulation of chloride at times and not sodium, and vice versa. We'll see the
Right.
accumulation of sodium, but not chloride.
Yep.
And and it's interestingly enough, talk about other salts. One of the most common historic agricultural inputs is MOP, uriate of potash, potassium chloride,
Hmm.
by which the plant's going to uptake it in copious amounts because it uses a lot of potassium and does not use large amounts of chloride. And so we see the accumulation, I believe, of chloride in the soil over time. And of course, when the plant takes up potassium, leave chloride behind in the soil, we have to assume that it's going to try and make partners with another cation out there, most often, calcium or magnesium, tie up in the
That's right.
calcium chloride or magnesium chloride, which in any rate is cement, of sorts. It's rock hard. It does not give you any of the attributes you're looking for in your soil for water infiltration or soil respiration. Um, but
You're
so
exactly correct. Exactly.
could you dive a little bit more into that? Because there's two things you're saying. You're saying when dry conditions arise, they must be present, or they are almost always present when mites become problematic. And then to that, though, you're saying when plants are become unhealthy. And I can't think of a single incidence where I could see a plant and say that's a healthy plant and it has a low Brix. So I think we're making a generalization that while there are varying levels of Brix in different plant species, uh, the lower the Brix, the more unhealthy the plant, and the higher the Brix, relatively speaking, um, acute events aside, because I have seen frost events or some even thunderstorms or things like that make Brix peak far above abnormal levels in response to stress. But
Or
all that
dive. Storms actually cause it to dive.
or
Yeah.
dive. Okay. But with that, when you're saying an unhealthy plant and low moisture conditions, but you're saying, okay, we often see low moisture in the plant when there's a cessation of rainfall events or irrigation,
Yes.
but you're not speaking selectively only to when the environment has little water. You're speaking to plants that are dehydrated,
At a
right?
cellular level?
At a cellular level. Is that
At a
correct?
cellular level, you are correct. Yep. Yep.
And now this kind of, maybe we can go there. Maybe this is a good segue because we were discussing earlier. We talk. There's a lot of talk about electron swaps. We talk about electrons a lot, and when we're talking about compounds or elements, and but something that doesn't get talked about a lot as much, I think, would be protonation and And there's a number of ways to look at that. I think there's the more traditional, conventional way to look at that's from the chemistry perspective, which is we have cations and anions, or we have molecules which have a net negative charge or a net positive charge. And those, but even so, we tend to really be focused, most people, I think, on the electron state when we view those charges. But there are other people I've talked to that prefer to discuss things in the terms of charged or discharged. They see those as diametrically opposed, rather than being able to have a negative charge or a positive charge. And we can set that aside because I think it's probably not very productive, other than for interesting conversation theoretically. But saying all that and talking about hydration levels in plants, we were discussing just earlier about this topic. It makes me think of what you mentioned, which was Dr. Gerald Pollack and the phases of water. And so it's something we've observed, that oftentimes plant health and plant hydration, is more often than not correlated to environmental stress. But there are instances, and we see them often, where they're not correlated, where we have dehydrated, if you will, drought conditions outside the plant, but hydrated plants, cells that remain hydrated. So I'm interested in what direction you might go concerning mites or not concerning mites in that.
Okay. Based upon what you just said, imagine, if you can, let's see, a cell. You understand what a cell wall is, and that it doesn't collapse, at least not normally. And you also understand that the cell membrane, when it's filled up like a water balloon, the cell membrane takes on the shape of the cell wall. So you're with me there in this visual that I'm trying to create. As the water is removed from the cell membrane shrinks. The cell wall does not. Now we have a disassociation between the cell membrane and the cell wall. Gerald Pollack has talked about, with easy water, he has talked about the accumulation of this easy water, which is essentially water in sheets, and that is very good at expelling all other types of solutes, is associated with membranes. This is what a healthy plant likes to have. This battery system that Gerald Pollack has talked about allows the plant to do what it is supposed to do. It is able to detect its environment and act in a quote-unquote healthy way. Once you disassociate the cell membrane with the cell wall, we no longer have that thin layer of water in between the cell wall and the cell membrane. Instead, you may just have a globular of water, because sometimes the cell membrane is still touching bits and pieces of the cell wall, but it's not filling it up like a water balloon would. And therefore, you have effectively changed the morphology of the internal parts of the cell. So therefore, it is no longer able to detect. Its sensory system has been compromised, in part because sensory system has to do with that the apoplast. The apoplast is a high concentration of calcium, and when we have calcium influx in a plant, this is considered to be the rudimentary sensory system of the plant. We have a similar type of system in animals where we have calcium influx. We measure a sensory effect by calcium influx. We can also measure a sensory effect in the plant by calcium influx. Once we have that water dissipate and the cell membrane separates, we have a breakdown of that sensory system. The plant is no longer able to do what it's supposed to be doing. And so already the physiology and the morphology of the plant has been compromised. When you have this compromising situation, it is very reasonable to assume that we do not have a fully functioning plant. And if it's not fully functioning, it's not going to be photosynthesizing at a full level. If it is not photosynthesizing at a full level, there is going to be less photosynthate produced, which is the end product of photosynthesis, which is sugar. If you have less sugar, you now can measure that with the Brix reading, because the Brix reading is going to take a look at the total dissolved solids, the predominant amount of which is sugar. And so if you have less sugar, you have a lower Brix, which means you have a less healthy plant. And this is based upon, unfortunately, a large complex of factors that I've just outlined to you, hopefully in the most visual way that I could, since I don't have graphics to describe it. And you can start to understand how the plant has become compromised through this particular system, which is the whole system is now more attractive in multiple ways to the two-spotted spider mite.
Wow, that's excellent. Thank you for that description. That paints an incredibly graphic picture, and it's got my mind spinning about it, thinking about it specifically, as you said, because so one thing that I thought of, you said it disconnects itself, basically disconnects from its ability to sense, and in so doing, you could argue that the cell becomes isolated from other cells and from its environment.
Yes.
And that made me immediately think of Dr. Zach Bush and some of the research he's doing on cancer treatment. And some of the latest research is suggesting that oftentimes what determines a cell becoming cancerous is a disconnection from the general species. Of course, we're super organisms, and so but for whatever reason, when a cell becomes isolated and disconnected from the superorganism, it often becomes rogue and has uncontrolled growth. And so I'm just seeing parallels and mirrors across different modalities. But so in this scenario, okay, the plant becomes dehydrated and the cell membrane separates from the cell wall, leaving air, and air doesn't frequencies don't move through air as well as they will through the solid. Is that right? Is that
That would be partially correct. So if the whole cavity was filled with water, you would have unevenness before the water balloon fills it. So you have a nice thin layer between the cell wall and the cell membrane. Once it separates, if it's filled with water, you now have a gap. There's like an ocean in between, meaning it's not operating the way that it should. So that's one of the things. Now, if there's air in there, as you've just suggested, that complicates it again. That's another complication because now we've got another breakdown of the whole system, especially that communication between the cell wall and the cell membrane and the apoplast, which connects the entire plant, because the apoplast connects the entire plant. And so you are unable to detect and send messages the way that you should or could do if a plant was operating in the way that it should. So yes, I don't know per se whether or not it's all air or whether or not it could be water, but either way, it's wrong.
right?
Plants should not have that big vacuous bag of air or a big vacuous bag of water. The only vacuous bag of water it should have is in the central vacuole, which
Right.
is inside the plant cells. That is there, and it's supposed to be there. But outside of the cell membrane, it should not be there. And yes, things start to break down. The plant's health has now become compromised.
So with that being said, do you have an idea what compounds, back to the spider mite issue, what are they after? What is their nutritional requirements? Why are they there at that time? Is it just the lack of water, or is it that combined with an increase of other compounds that they're after from a food source? And with that question,
Yeah. What's
Yeah.
in it?
why are they different, or what are they after, for example, that say something like an aphid is not?
Big question.
Good question. You're right. I just mentioned really the first step, that dehydration step. Now you've got to have something that is fundamentally attractive for the mite in order to eat. So another thing that happens in the plant when we have excessive heat, so we're having 95, 100-degree temperatures, the plant starts to dehydrate. We've already set that scenario and we've already taken it maybe as far as we can. Now we've got another problem. The plant starts catabolizing itself. And in the process of catabolism is a sense of breaking down its proteins as it starts to, in its relative state of unhealth, it starts catabolizing itself. And you now, instead of having complete proteins moving around the plant, you now have broken down proteins moving around the plant. As this catabolism process occurs, these broken proteins become quickly more numerous. They cannot easily go back and create complete proteins again, which means usually what we find is that the two-spotted spider mite can feed even after water is restored because the whole plant has already started to break down. But these catabolism products, which essentially would be, let's call them small proteins like dipeptides, tripeptides, pentapeptides, can be detected by their peptide bonds. So one of the main frequencies that insects have when they are attacking plants are the peptide bonds. They cannot detect the peptide bonds easily in a complete protein because you might have 400, 500, 900 amino acids in a particular protein. Therefore, you're always going to have, if you have 900 amino acids, you are going to have 899 peptide bonds. Well, that's a very complex message right now because the protein is so complicated that the infrared message is going to be equally complex. However, when you take a protein and break it down to the point where it's now just a dipeptide, there's only 2 amino acids, the peptide is easily picked up. And a dipeptide bond can be easily detected with equipment that we have, especially like FTIR and some of the other equipment that we have. And because it's detectable, not detectable when it's a complete protein, but detectable when it's broken down, this means that maybe, just maybe, the insects and the mites are able to detect these peptide bonds, meaning they're able to detect the broken down proteins. And we found it. We found the frequency for the dipeptide bonds in the aphids, and we found the same frequency in the two-spotted spider mite, meaning, to answer your question, Trent, and I realize that I'm going in a long, circuitous way, but I'm trying to teach while I'm talking, is that we have now a mechanism where the insect, the mite, sorry, the mite is now going after these broken down proteins. It detects them, it wants them, and these broken down proteins are taken into the mite and then formed into its own complete protein so that it can make all of the proteins that make up its own eye, leg, digestive system, cuticle, whatever it is on the mite that needs to be made, it can be made from these broken down constituent proteins, these broken down proteins, after the plant starts catabolizing itself. How am I doing so far?
That is awesome.
Blowing our minds, I think. So when you say these dipeptides, like what is in your estimate, what's the best way to detect these? Is this through a plant analysis, or can we do this through some sort of frequency, non-visible spectrum that we can utilize?
Woo, that's actually beyond my pay grade. So the way that I discovered this is through a circuitous route, and that is by taking a look at the actual chemosensory proteins, which include things like odorant receptors or ORs, gustatory receptors, GRs, ionotropic receptors, IRs, as well as I'll also take a look at the SNMPs, which are the sensory neuromembrane proteins, as well as some other proteins. And so when I break down these proteins in an aphid or in a two-spotted spider mite, I scan them and look for the predominant frequencies that emanate, knowing that a predominant frequency is what they're keying in on. And they're not going to be focusing on something that's a smaller frequency because I'm looking at a cumulative effect of all of these proteins together. And when I take a look at the cumulative effect of these proteins, one of the predominant frequencies is the dipeptide frequency. So to answer your question, what I'm trying to say is that they are able to taste the dipeptide frequencies.
Wow.
Am I still blowing your mind?
Yeah.
So it's a little bit different than what you're thinking of. So can we detect it? And we have ways of doing that, but the two-spotted spider mite is actually going to be sticking its mouth parts into the dehydrated cell. It will start sucking, trying to make sure that it has what it's supposed to be eating, which is a lack of water. It can't take too much water. 2, it's got to have these broken down proteins, and if it is, it will continue to suck out the contents of the cell, and then it will move on to the next cell, which causes that stippling, that spotted effect that we see with the two-spotted spider mite, which is so indicative of its feeding because it cleans out a cell, removes its mouth parts, and then goes to the next one. And hopefully, for its own sake, it finds the same thing so that it can continue to start cleaning out the contents of the cell. And then when the numbers get to be very high, such as 1,000 or even 10,000 on a particular leaf, you have the leaf essentially being evacuated of all of its nutrition, and there's really nothing left. And at that point, proteins. Photosynthesis is just not happening anymore because they specifically go after the photosynthesizing cells, the mesophyll layer. They don't go after the epidermal cells. They go after the mesophyll cells, which are the ones that contain the chloroplasts. So because they're evacuating specifically the cells that contain the chloroplasts, there's nothing left for the leaf to photosynthesize. And the leaf essentially turns a light green. Sometimes it turns a bronze color, but essentially it's lost its ability to photosynthesize if the majority of the cells have been evacuated of that mesophyll layer, where the chloroplasts are located.
So do you see a healthy, what does it look like in a healthy relationship? I mean, where the mite is not decimating the plant, where, I mean, I guess you'd have to first assume that they're healthy plants, because when there's unhealthy plants, then the mites are able to have a feeding frenzy. And as there's an abundance of ideal food for them, they're going to reproduce, and then their offspring will have abundant food, and conditions will be right for them to reproduce, and so on and so forth. But they are a natural part to the ecosystem. And has there been any beneficial effects of mites found, or what does it look like in a functional relationship where mites exist with plants, but not to a crop detrimental effect or impact?
Okay.
What would that be?
Well, that's kind of what I mentioned a little bit earlier on, and that is if you go into any field, whether it be Washington, Florida, or anything in between, you will find two-spotted spider mites, meaning they're there and they're doing what they do, which is sucking out the contents of particular cells. Most of the time they don't do it well. They need a particular cell in order to evacuate the contents of it, and if the plant is generally healthy, they're going to have to start being very, very selective about what cells they evacuate the contents from. Meaning this is going to take time. It's a slow process. They're not going to grow that fast. They're not going to be able to produce the young that they need. It's just a very slow, inefficient process. But they're still there. And they can still suck out the contents. They just don't do anything to really affect the plant in any serious way, because we're talking about thousands and thousands of photosynthetic cells just within one leaf. And the fact that some trees have thousands and thousands of leaves, this makes things much more complicated for them to overcome.
Thanks so much for joining us as we expand our paradigms and explore what's possible in the realm of regen agronomy. If you would like to contact us or learn more about what we do, please visit our website at www.soilcraft.com, as well as our YouTube channel. If you have topics you would like us to unpack or stories, please connect with us so that we can share them with others.